So Chris, tell me what your research is on.
Well, we're working on climate change, and we're trying to project forward and see how the conditions that are going to occur in the future will influence Diamondback terrapins.
And why might climate change affect Diamondback terrapins?
Well, terrapins, like most other animals out there, are cold-blooded, and meaning that their body temperature responds to the temperature around them. And that means a lot of the functions within their body are temperature dependent as well. And something that's special about many turtles, including the Diamondback terrapins, is that during development of the embryo, it's the temperature of the nest that determines whether or not the embryo develops as a male or a female. So the temperature of the nest is very important to the population as a whole, in a sense, because it determines how many males and how many females ultimately get into the population. Now, of course, with climate change, temperatures are changing. Therefore, the temperatures of the nests are changing as well, and they're getting warmer. So part of our work is to monitor the nests in nature, to go out there and catch the females while they're nesting, place data loggers in their nests with the eggs, and record the temperatures of the nests. And then over time, we can develop a database and see how the nest temperatures are responding to changes in the climate overall.
And do you bring terrapins into the lab to work on them here at CBL?
We do. We bring terrapins in for several purposes. Sometimes, depending on the study, we would bring them in as eggs and we will incubate them. And by incubating them in the lab, we can control the temperature at which they're exposed during embryonic development, and therefore we can ask questions about how development of male and female traits responds to temperature.
Can we look at those incubators?
Sure.
All right, so here we are with one of the incubators. Tell us how it works.
Well, this incubator has heating and cooling elements in it, and basically it allows us to control the temperature every minute of the day, throughout the day, in order to mimic the temperatures that we see in natural nests. So we have little computer controllers that are reading data from the nests that we measured in the field last year and using those data to set the temperatures at which the nests will develop in the incubators. What that does is that allows us to mimic the conditions that are occurring in the field, but at the same time make certain manipulations to those conditions. For example, with this study, what we were doing was we were looking at whether or not the temperatures, if temperatures come up 2 to 3 degrees warmer or were to be 2 to 3 degrees cooler than the average temperatures, what would that mean for the development of the animals? And so we look at the growth of the animals after hatching, but we're also interested in whether or not they can out.
So let's look inside one and see what is going on.
Okay, so in this incubator, we have our eggs. Now these eggs have been incubating for close to 2 months. They've been at a fairly cool temperature, so they haven't hatched yet. Other eggs that were collected at the same time but raised at only a couple degrees warmer temperature have already hatched. And we'll look at some of those hatchlings in a minute. But these are what the eggs look like, and you see each egg has a number on it that we wrote on it with number 2 pencil when we were collecting the eggs, so we know which egg came from which nests in nature. One of the things when we do with these incubations is we try to get eggs from a bunch of different nests into each incubator. That way we can start to look at questions of how do different clutches, how do different nests affect how development occurs.
And can we look at some terrapins that have just hatched?
Yes, we can. We have some right over here. When they first hatch, there will still be some yolk sticking out of the belly there. In fact, one of the things we do is we measure the size of that yolk when they hatch, and that gives us an index of how prematurely or maturely they exited the shell.
And how long ago did this terrapin hatch?
This terrapin hatched a week ago.
And let's see, how big is this terrapin?
Okay, so, well, this terrapin, most of the terrapins that we hatch from the local population weigh somewhere around 8 to 10 grams when they hatch. This is a fairly large one. We're going to weigh it right now, because one of the things that we do with all of the animals we bring into the lab is we weigh them and we measure them, and then we know the size of them when we release them later. If we were to recapture them, this animal weighs 9.4 grams. It's rather eager to be reweighed, I guess. We also then we measure their length. So the back shell of the turtle is called the carapace. So we measure the carapace length, which with a pair of calipers, which we measure from this end to this end, and I always try to keep its head pushed down there so it doesn't get pushed by the calipers. And we measure this one's 33.2 millimeters long. I think I got that right. And we also then measure the width of their carapace. So, and that is a measurement like this. And we measure that at 30 millimeters, 30.2 millimeters across. So now we know how much this terrapin weighs, and we know how long it is. This terrapin now will be marked, and we'll take it back to the field where the nest was collected, and we'll release it.
How on earth do you mark a terrapin?
Marking terrapins, especially terrapins of this size, is very tricky. One of the issues with these turtles is, of course, they're small, and so it's very hard to mark them because of their size. But another issue is that as they grow, depending on the type of mark you make, the mark may not stay with them. The best way to mark animals in the wild that we've found for our work is to actually insert a transmitter into under the skin. But of course, on an animal this size, that requires surgery, and that's very difficult and time-consuming. Rather, what we've done is we've developed a technique, which I'll demonstrate in a minute, where we actually put a tattoo mark on the plastron of the baby turtle. Now, we can do that on these baby turtles because shortly after they hatch, and for probably a week or a little longer, their shell is quite soft. And so the tattooing needle will pierce the skin just like a tattoo on a human and leave the ink behind. The idea is that these tattoo marks, we hope, will stay with the animals for several years. We don't expect them to be there for the lifespan of the animals, given that they can live to be 40 years or more, we think. But what we do hope is that the tattoo marks on the plastron will be around long enough so that when we capture them as adults, we can remark them with a more permanent method.
Can we see a tattooed turtle?
Sure.
So explain what you're going to do.
Okay, so we are going to mark this hatchling. First thing we're going to do is we're going to put a little nick in the shell, right up here where these two parts of the shell come together here. We call these marginal scutes. Where these two scutes come together, we'll just put a little nick in there. That way, and that the animal will retain theoretically for its whole life, although it will become harder to see. But what that will do when we find the animal that has that mark is we'll know that we're going to need to closely inspect its plastron for tattoo marks. And again, once we've identified it that way, as the animal will be an adult at that point, we'll be able to mark it with a more permanent method. The other thing that we've been doing is I'm cleaning this with alcohol. That's why I'm daubing it off there. Is we're also saving this small portion of the shell for future DNA work, or possibly future work on the digestive or the feeding ecology of these animals using isotopes. Regardless, at least
that's a small piece of shell that you take away.
it is a very small piece, very small, almost not noticeable. However, it will provide a little indication of asymmetry on that part of the shell that we'll recognize as being a previous mark, and then we'll know to inspect this side. In order to tattoo these, we first clean the plastron, and then we have the shells divided into plates or scoots. And there are 3 of these that we specifically mark. It's this, and this, and we have a pattern. It's a combination of letters and numbers that indicate where the animal was captured, the year that it was captured, and the animal number. So we'll place a letter indicator here for the location, a number here for the date, 2020, and the number here for this animal ID. This animal is ID number N2037. We'll get an N, a 20, and a 37. So let's see how it goes.
And this is just the same sort of tattoo machine that you would find in a tattoo parlor.
It is. This is off the rack, Amazon, make your own tattoo parlor kit, I guess. So we have the end, and I'm sure it's hard to see given the pattern on this animal. That's why we have this mark on the other side, because we recognize that this is going to take considerable visual inspection to identify these animals. Now we're going to put the 20 on this scoop. So there's mark number N20. Make sure we've got plenty of ink. And finally, this number 37. And that's a tattooed animal.
So there we have it, N2037. Chris, we've now seen N2037 being tattooed. What happens to this terrapin now?
This terrapin will go back to the site where the eggs were laid, and it will be released. And we will hope that several years from now we may recapture it when it comes back to nest.
And when will you know whether it's a boy or a girl?
Well, Diamondback terrapins are fairly slow to mature. If it's a girl, we may not see it again for 6 or more years. If it's a boy, we may see it again in 2 or 3 years, maybe a bit more than that. The males don't come on land to nest, so we rarely capture them unless we're specifically looking for them through trapping. So chances are N2037 will be on her own, or his own, as the case may be, for several years. And during that time, it's got to face a lot of hardships, and it's going to be a part of the natural system out there. It's going to be a an important predator on small animals, and it's going to be an important food source for other animals.
So your experiments take a long time. This isn't something that you get the results immediately.
That's a fact. One of the difficulties in working with reptiles, especially turtles, is that they live a long time, and it takes a long time to collect data on any individual throughout its lifetime.
All right. Thank you, Chris.
You're welcome, Tom.